morning again, and my name is Paul Wang from Radiology, and I'm going to share with you some of the imaging capabilities. The mission of this core facility is to promote and support and long-term research at Howard in using imaging technology. And basically, we're trying to use this imaging technology to study the mechanism of disease processes and their response to therapy at molecular or cellular, subcellular, cellular, or vivo level. And specifically, we are going to provide state-of-art instrument and technical expertise to serve the community for the molecular and cellular in vivo imaging. Also, we'll provide the training for research if you want to use any of this imaging technology. And we, of course, as a center, we want to foster the research collaborations and anything we can help to help you in your research and to link the different people at a common interest. So this is what we are doing, what we try to do. The structure of the imaging core, basically there are two parts. This is an ultra-structured imaging core, and my lab is a medical AMR and molecular imaging lab. In the ultra-structured imaging core, we have Dr. Masari in charge of electron microscopy, and Dr. Olson is in charge of photo. And both Dr. Masari and Dr. Olson going to offer me a brief introduction of their imaging capability. So under the biomedical AMR and molecular imaging lab, we have three functions. First, molecular cell biology, try to understand the underlying mechanism of some imaging processes. And also we have a probe design. We have a chemist, we can design some probes for imaging purpose. And of course we have imaging capability, which involves MRI and optical imaging, which I will explain in more detail. And these are the following are the people in my lab. Here's Sophia, our research assistant, and Stephen, and Dr. Zhang is a biologist, research scientist from China, and Dr. Schiller is a radiation biologist from radiation ecology, and Alex, Dr. Korikov is an imaging scientist, and Dr. Olson, it's not my lab, and she's in charge of the control. And Dr. Ye is a chemist, who's the person in charge of designing probes. And Dr. Sarin is going to join us shortly. He's a neurosurgeon background. And first, I would introduce one of the two imaging capabilities in our lab, which is optical imaging. Optical imaging, basically, you can see the foreground in the animal. So everything we do in my lab is small animal imaging. So there's we do not do we don't do patients. So optical imaging, we have an instrument called Xenogen by Xenogen. Basically, it's a black box. You can put a small animal here, and you can shine the light, and for you need the light. You can detect here it is. So you can put a small animal here inside the box, here, and you can generate basically two types of images. This is a fluorescent image. So basically you can see chlorophyll and see where it is. Or you can see luminescent image, bioluminescent image. So the cells, we can transfect cells with the receiver's genes, for instance, and you can image where the cell is. Just one slide for optical imaging. Luminescent, bioluminescent. Basically, here I use an example. See, breast cancer cells, which metastasize into the lung, And these cancer cells, which has a luciferase gene, will produce luciferase in the cells. And if you inject luciferase IP injection, which will oxidize and emit light, so you'll see where the cells are located. And this will be very useful for the research if you want to trap some cells, for instance, stem cells, or you can see the metastasis, cancer metastasis processes, and all this is done in vivo. And according to the company, this technique, you can detect fewer than 100 cells in vivo. So that's depends on, of course, depends on how much the light will emit from these cells. And optical imaging basically is extremely sensitive and very low cost compared to CT, MRI, and PET scans. And down the road, there is tremendous potential for clinical use. And this is an example of head and neck tumor here, and you can easily detect the tumor at a different time. So this is a non-invasive methods which you can study the processes or to monitoring the therapeutic effect, so forth. So it's kind of useful for longitudinal studies. And the second thing I want to introduce is the MRI, the imaging capability of MRI on MATLAB. MATLAB, we have two MRI machines. So one is 4.7 Tesla machine, the other one is 9.4 Tesla machine. Versus a clinical machine, normally people use 1.5 Tesla, or sometimes people more advanced use a 3T Tesla. So we have two very high power MR machines. And this 4.7 Tesla machine is horizontal board, which you can put a small animal horizontally. The largest animal we can put in is 20 kilo small pig. So this is an image throughout the rat brain. A couple years ago, we worked with Dr. Manet, use Alzheimer's disease animal model, which we can easily monitor the brain volume, hippocampus volume, and so forth. This image is an image of the lung, of the mouse lung. And as you can see here, there are two tumors. And the small tumor here on the left, which is about the size of 2 millimeters. The imaging capability of these systems, we can get down to the spatial resolution close to 50 microns. The pixel size is about 50 microns. So here's another few examples of the imaging capability. This is a sagittal view of a mouse of the prostate gland. This is bladder. This is the spinal cord, so you can see the orientation. It's a sagittal view through the midline. And this is prostate gland. And this is mouse with a prostate tumor. So this is the tumor, this is prostate gland. So imagine this is about 20 gram mouse. And also we can do cardiac imaging. This is image of heart. And you can synchronize your MRI data acquisition with cardiac cycle. And so you can make a movie out of this. So you can avoid the artifact from the motion object. Or you can, the image on your right is an image of angiogram, of a mouse angiogram. Two branches of carotid artery coming out from the neck to the brain. So again, this is mouse brain, and you can see the resolution. So this show demonstrates the imaging capability of MATLAB. And this is another image of in vivo mouse brain and image of spinal cord, a live animal. And this is from the specimen. And from the specimen, actually you can keep this specimen there for a long time. So you can push the resolution. Or in vivo, usually we can keep the animal in the machine for 2 hours. So within 2 hours, you can do whatever manipulation. Besides imaging, we can also use NMR spectroscopy, study the biochemistry of the tissue. Here I give you an example. This is a a sectional view of mouse muscle. We have a collaboration with the children's people. They are interested in the muscle dystrophy. And the type of information you can get, we can take an image first and zoom in particular part of the body. Here, we zoom in the muscle. And actually, you can get a series of spectra at a different time. This is an example of we start the blood supply of the muscle. So this is a phosphorus spectrum. So what you see here is 3 ATPs, alpha, beta, gamma, and phosphorus creatine, inorganic phosphate. This is before you clamp the muscle. Now, after you clamp the muscle, immediately the system will use up the ATP and PCR and so forth. You can monitor the drop of this each compound. And after a while, you resupply the blood, and you can see the recovery of this. This is just simply an example of what you can do using our machine. You can see the biochemistry non-invasively. At the same time, you take an image. So the focus area, research area, we're doing small animal imaging. We use a lot of tumor model, breast, prostate, melanoma, head and neck, and so forth. And it's very good for anatomical, functional imaging. For instance, we can use it for Alzheimer's disease or cardiovascular diseases. And this will be useful for drug development, translational studies. We can do in vivo drug metabolism, study the pharmacokinetics. Like a NMR spectroscopy, you can see the different compounds and you can study the pharmacokinetics. And in the last few years, we have developed some nanoparticle for therapeutic or diagnostic purpose. So we are doing a lot of research in that part. Neuroscience imaging, Dr. Sorry, in charge, and they are doing a lot of study disorders, mental health disorders. And other than that, I'm working on the central peripheral nerve systems. And also, we are interested in the mechanism of this nanoparticle getting to the cells, how this getting to the cells, or the safety issue related to nanomedicine. So this is a kind of a current research area we are doing in the lab, but this is not. We are not limited to this. If you have some other interest, we will actually talk to you and see if we can help. And just to give you a few quick examples of what the research we are doing. Here, first, we developed some nanoparticle, use transferrin receptor as our target. Transferrin receptor. Suppose here is the cells, inside the cells, in the cell membrane. Probably 70 to 80% of the cancers overexpress transferrin receptors. And we use that as our target. So we can develop some nanoparticle. Here we use liposome. On the surface of liposome, we can put some ligands, like antibodies or antibody fragments, which we can targeting transferrin receptors. And through endocytosis, and you can carry whatever the payload inside this nanoparticle to this target. So in our case, we developed this mechanism for diagnostic purpose as well as to deliver therapeutic agents to the cancer, to the tumor. So here's an example. We can, on the surface of this nanoparticle, we can put a transfer label with fluorescent dye. So this is a mouse, and this is tumor, and this is before we inject the probe with the fluorescent dye, and after 10 minutes, 30 minutes, so forth, actually you can see this probe accumulate at the tumor site. So you can use this for detection purpose. And likewise, inside of this liposome, we can put MRI contrast, image contrast agent. So we can selectively deliver MRI image contrast agent to the site. Here is an example of mouse tumor. This is a cross-sectional view of the mouse through the abdominal region. This is the back of the mouse, and this is spinal cord, back muscle, intestines, and this is the tumor. Okay? And we have two cases. One is MRI contrast only, the clinical MRI contrast agent, before and after inject to the animal. And this is with our nanoparticle for targeting before and after. So you can see the difference between use conventional MRI contrast versus use this targeted delivery. As you can see, shows tremendous heterogeneity inside the tumor. And also that reflects the targeting process, because we're see the mechanism is seeking through the transferrin receptors. For cells which are fast proliferating cells, which has a lot of transferrin receptors. So actually you can use this for prognosis purpose. So if after treatment, if the cells are dying, you will see less or fewer number of transferrin receptors, or the other way around. So actually you can use this for prognosis or monitoring the therapeutic effect. Then another example, just very quick, we can use this nanoparticle overcome cisplatin resistance. Cisplatin is one of the major drugs used for cancer patients, probably the first drug we use. However, it's very effective at the very beginning, but a lot of times the patient develops resistance. So we try to use this nanotechnology to overcome this resistance issue. The thing we are developing is to use a nanopolymer, which has a carbon 82 carbon wall. And on the surface, we can put some ligands for targeting purpose. And this is just an example. Okay. We have 4 groups. This mouse, on both sides, we develop tumors. Say on the right side, we have the cancer cells, which is drug cisplatin sensitive. On the left side is cisplatin resistant. So you give the mouse the cisplatin, the sensitive will shrink, the resistant will stay. Okay? With nanoparticle, doesn't affect too much at all. With the cisplatin plus nanoparticle, both sensitive and resistive tumor shrink. This is the detailed mechanism published in ANS this year, and you can look it up. But anyway, this is just a quick example of nanoparticle, and in this particular case, for animal, at least, you can see which overcomes cisplatin resistance. And on the right, that's the control. And you can also use these optical imaging methods for fast screening process. And you can 96 well cells, you can edit all kinds of different conditions, and you can see their emitted light, which reflects some biochemical processes. And we can use this for some study of the pathway. In this particular case, it's a breast epithelial cells, and which has growth medium, and we have some transfect with the beta-casein here. We have can emit light, and actually we can study all these pathways by varying the different conditions in the cells. So you can, from the intensity of the cells, you can reflect the pathways. So these are the fast screening methods. I'm sorry, we don't have too much time for the presentation. If you have any interest, maybe after Dr. Postman, Dr. Nasari's talk, you can ask me.